Click the bell icon and subscribe to our channel so that you will not miss any classes. Now we shall discuss application of biotechnology in the field of medicine. Medicine, it always deals with the treatment of particular diseases or disorders. For that, we need some therapeutics. And those therapeutics, some of them are isolated from non-human resource. So, non-human resource generated therapeutics are found to be generating some unwanted immunological response in human beings. So, which is a disadvantage. And hence, biotechnologists have thought to produce a recombinant therapeutics. That is, the major application of biotechnology in the field of medicine is for the production of recombinant therapeutics. And worldwide, there is production of 30 different recombinant therapeutics on a large scale, and 12 of them are being approved for human usage in India. You need to remember these, and it is important for competitives. And to name some of the recombinant therapeutics which are produced, for example, insulin, human growth hormone, interleukins, fibroblast factors, so on. Many types of recombinant therapeutics are being already produced using recombinant DNA technology. Now, in this class, we shall talk more about the recombinant insulin. To understand that, first we shall understand the physiology of insulin, where it is produced and how it is processed, or what is its function in human body. Insulin is a proteinaceous hormone produced from beta cells of islets of Langerhans. And this insulin has two roles. When it enters into blood, it stimulates conversion of glucose into glycogen. Process is called glycogenesis. And secondly, it acts upon hepatocytes and adipocytes. Hepatocytes means cells of liver. Adipocytes means cells of adipose tissue. And when it acts upon these tissues, the tissues will uptake and utilize glucose. So, now what is the function of insulin? Insulin is a proteinaceous hormone produced from beta cells of islets of Langerhans, and insulin has two roles in our body. First one, it will stimulate conversion of glucose into glycogen. The process is called glycogenesis, and also it acts upon the cells of liver, hepatocytes, and cells of adipose tissue, that is adipocytes. And when it acts upon these cells will uptake and utilize more amount of glucose. So, glucose level will be under control by these two. So, generally, in order to say generally, insulin is majorly responsible for maintaining the blood glucose level. If insulin production is decreased due to genetic factor or due to age factor, then glucose level in the blood will increases and the condition is called hyperglycemia. Or diabetes mellitus. And this diabetes mellitus condition can be treated by introducing or artificially injecting the insulin into the blood. For that, there are two approaches. In earlier days, in a conventional method of treating hyperglycemia, one used to isolate. The first approach was developed in order to treat the insulin was they used to isolate insulin from slaughtered animals like pig and cattle. And when the insulin was isolated from non-human source, then that was developing a unwanted immunological response. That is, insulin isolated from slaughtered animals was found to be allergic to some patients. And also one more problem was amount or quantity of insulin which was isolated was too less and it is not sufficient to treat large number of patients. For that, scientists have thought to introduce insulin. Second is biotechnological approach. The second approach was biotechnological approach. In biotechnological approach, scientists have thought to introduce insulin gene from human beings into bacteria so that insulin can be produced on a larger scale. Since it is from human gene, it may not induce any immunological response. So to understand biotechnological approach, we need to study the structure of insulin first. In human beings, insulin is produced in the form of proinsulin. Proinsulin, it is inactive form of insulin. That is, we have discussed already protoxin, proinsulin. That is inactive form. The inactive form of insulin contain 3 polypeptide chains. They are namely A chain. So this is A chain, which contain A chain. It contains 21 amino acids in it, and B chain, it contains 30 amino acids, and C chain, which is containing, this is C chain, between A chain and B chain there is C chain, and C chain contain, this is C chain, it contains 58 amino acids. This is the inactive form of insulin. Now, when it is released into blood, there it is get activated. How it is activated? By the removal of C chain. When C chain is removed, the proinsulin will become active insulin. Active insulin means it contains only A chain and B chain. Observe clearly, I am writing A chain little shorter and B chain little bigger because the difference in amino acids. Okay, here only 20 amino acids, here 30 amino acids. That's why. So now these two chains are linked by disulfide bonds, and now this insulin is called, here also disulfide bonds. This insulin is called the active insulin, which can stimulate the conversion of glucose into glycogen. Now, what scientists thought is, only they have isolated the cDNA. cDNA means, you need to understand how they isolate cDNA. They were interested only to isolate A gene, the gene coding for A chain and the gene coding for B chain. There is no use to produce the C chain because it is, whenever it is present with insulin, the insulin will be inactive. So to produce active form of insulin, they need to isolate only A chain coding gene and B chain coding gene. Now what they did is, they have isolated the genes coding for A chain and B chain. Remember here they have artificially synthesized the cDNA. Here I am telling directly the gene. Actually it is not the case. Here what they did is they allowed the genes to express. Gene coding for A chain and B chain were allowed to express. After expression they will produce mRNA, right? So here also it produces mRNA. From this mRNA, they synthesized the DNA artificially. So this DNA is called cDNA. You may ask why? Why? Because this gene has to be introduced into bacteria. We know that bacterial genes are polycystronic, whereas the eukaryotic genes are monocistronic and they are intervened by exons and introns. So, whenever they directly isolate the gene, it contains both exons and introns. So, which is a complicated machinery to express in the bacterial host. That's why they first isolate the gene coding for DNA coding for A chain, B chain, allowed it to express, and after production of mRNA, from that mRNA, cDNA of A chain and B chain were synthesized. Then these two are attached with the plasmid of Escherichia coli. Plasmid. From here onwards, the process will be same. But process is same, but they will be conducting separately. Okay, it will be attached with plasmid having beta galactosidase. Then they were separately introduced into E. coli bacteria, two cells, E. coli bacteria. Now in one type of Escherichia coli bacteria, there is expression of A chain. A chain will be produced and it contains or it is attached with beta galactosidase. B chain was also attached with beta galactosidase. Then these two were treated with a chemical cyanogen bromide. Then pure A chain and B chain were obtained. Then these two were mixed so that there is a formation of disulfide bridges, that is A chain and B chain are connected with each other by a disulfide bridge. Now, what is required? A chain and B chain together. Together it is present, that is active form of insulin. It can convert the glucose into glycogen. So, it is ideal for the usage of human beings, and such insulin which is produced using a recombinant DNA technology is called Humulin. So, for 1 mark and all, they may ask you, what is Humulin, like that. Okay, so insulin produced from the recombinant DNA technology is called Humulin. Okay, so this procedure was followed by an American company, that is Eli Lilly in the year 1983. There onwards, there was a drastic improvement in the treatment of insulin. Till today also, the insulin produced from the same process is available in the market. So that is a one huge application of biotechnology in the field of medicine. From next class, let's continue with the other applications of biotechnology in the field of medicine. Okay, thank you.